I'll turn on the back side.
Yeah, it worked. We've got some data. Surprisingly enough, plate one was a full plate of 95 specimens, and it hasn't worked very well. We've got something like 20 or 30 sequences working. That plate two, which was incomplete and had 50-something samples, most of those have worked. So that's almost certainly just a specific organisms that were actually on it, because plate one was mostly marine. Yeah, we've got the pictures here. Plate one was mostly marine taxa. Like there were a lot of crustaceans, a lot of mollusks, which tend to need better quality DNA extraction methods, in my experience. And also the primers we used were probably not particularly optimal for those. These primers were kind of optimized for us. So then plate two, yeah, you can sort of see it stopped here. And the color is giving you a read count as well, like the number of reads that actually came from particular specimens. And then the darker is basically it's more reads. So we're kind of got very few reads here, a lot of reads here. So so we've got there's results. We haven't really looked at it in a lot of detail yet, but you guys are certainly going to have something to look at here. Yeah, we've got this is the read retention. So we talked about that's about 140,000 reads, like raw reads at the start of the run, and then every step that it does includes the laser pointer. So the first thing is you do size filtering. So I don't know how many of you got a chance to look at that, the read length histogram that PONT shows you. So it was like, if you saw it, it was like a peak that was at about 700 base pairs, but there were also these long-tailed of very short reads and very long reads. So that's just sort of artifactual data that you always get with the 90 short reads, maybe because a read went through the core and just get completely red, and long reads maybe because of concatenars that were multiple molecules that got stuck together, which in both cases are just sort of junk that you have to throw away and can't really do anything with. Demultiplexing. So demultiplexing means, of course, recognizing the UNI sequences, so we're able to place a read to a well. So you can see we actually lost probably about half of the reads we had here. We just lost because the software was not able to find any UNI sequence. So again, that's probably just low quality data that we have to throw away. But we've still kept about 60,000 reads, which for only 2 plates, that's a decent number of reads. And remember that because we're using asymmetrical UNIs here, the software needs to see both the forward and the reverse read to unambiguously map what a read was to demultiplex. So that's kind of why it forces you to keep only better quality data here. Reads after primer trim, just a very, very small amount we've lost here, but again, we only keep reads where we can actually find both primer sequences, so we know those are definitely one. And it also uses the primer sequence to orientate the reads in the correct direction, so everything ends up in the correct 5' to 3' direction. Because when a read with Oxford Nanopore, a read could be in either direction. It could be forward or reverse. So we want to end up there. Then there's these clustering. So we're putting identical or nearly identical sequences into one thing, so the correct groups or clusters. Reads after the text is a funny size here.
Yeah, I don't know why. You can maybe see my
reads after we've managed to put an identification on something, and then reads that it's selected for what we think the row to use operations. Okay, so we go ahead here. The reads per specimen. So because a relatively small number of specimens actually worked here, the reads we have are really concentrated in those few specimens. So the actual average reads per specimen is really quite high. Most of them have more than 500 reads. This is like the success per plate. We got 21 successful sequences for plate number 2. For plate number 1, 21 things, so that's only like 20% success. And for this, we got 48 reads out of the 50-something specimen. So yeah, plate 2 worked much better than plate 1 in the sequencing. Most specimens So this is where we're talking about finding those non-target sequences, where you find extra sequences from a well, which might be contamination, it might be bacteria, or things like that, human, because we've sequenced ourselves. So there's a few things with 2 sequences per well or 3 sequences per well, but nearly everything where we've got a sequence, it was just 1 sequence per well, which is kind of go ahead. We've seen that one. The read count heat map. The order composition of the run. So this is much more mixed than we typically see, where you can do a lot of sex or occasionally lots. Yeah, so what's the biggest group? The biggest group of things that work is Diptera, that's the flies. Also quite a lot of Hymenoptera, that's the reconnants that we put in. Rodents.
Rodentia, o sea, roedores también. Murciélago. Ha salido bien. Ha salido bien.
Calanoide lo veo sin verme. Calanoide sin verme. Octópodas, bueno, sin verme. Terópoda también, terópoda también. Ajá. Octópoda creo también de marino.
Yeah, so the total reads which we actually sort of use from the run, we can assign to specimens, 42,000 specimens with, specimens with a sequence of 69, average read 600, 69 recovered barcodes, nothing in the control wells apparently, which is good, so that suggests we didn't have a lot of contamination. And then we get your table here with all your results. So this is all of the context, not necessarily just the ones.
I have a question. Yes, the map or the taxonomic assay is with public and private database.
Yeah, so this is done by ontology's like local copy of the Bold Distilled library, which it does include private data.
I think this one,
this version does.
does it? Okay. Wasn't sure which one.
And it's using that program called Syntax as a taxonomic matcher. Did you explain about Syntax in your talk already?
Not really.
Okay. It's similar to BLAST. It's a thing of matching, assigning probable taxonomy based on similarity. So this is not using Bold. It's using data from Bold. It's using Bold Distilled, which is like that reduced version of Bold. That's the minimum number of representatives.
But it's using just public data.
I believe the version that is built into ontology includes private data as well, anonymized, so you can get access to information about that data, but the sequences are. Yeah, so you just get this entire contact table, so it's one row for each sequence that we generated. So these are read counts. This is the number of times that we saw this sequence, so 3, 32, 50. The minimum that we will count is 5. We've set 5 reads as the minimum threshold that we'll allow for sequences. And as we say, it includes what we're calling non-target sequences. In this case, D11, the only sequence that we got is an NTS, a non-target sequence, which is a gastropod. But I guess in this case must be just because the order level ID that Syntax thinks the sequences is not what was supposed to be in that table. So it's possible that you have a misidentification in that case. So we do still want to look at all of these results. But by default, that will not be in the set that it will output for upload to Bold. So I think we can send all this to you guys. You can look through this for your own interest later. But
It worked.
Yep, and it worked. So well done, everybody.
And I think one group, Honey, Simena, Daniela, and Valia will present on the data tomorrow.
Also, it would be good if you could double check that data sheet that Jorge sent us, because there were what looked like a few mistakes to me. Firstly, Hymenoptera was misspelled. It was HIM or HY or something, which would cause an identification to fail because it's doing exact text matches. There were 1 or 2 rows that were missing any taxonomic information but did have like a specimen ID for your specimen, so maybe just double check what that should have been. I kind of guessed by copying the information from the row next to it, but maybe kind of go back and look at that again. Yep, that's all.
Yes.
So we're just working on more projects there.
Just telling Paul and Amina and Sean, CCU that we got the data.
Esta data no la
Rapid base
pasaba, ¿no?
calling on the market see. I'll tell you when to crash out and
Right.
die. So we should only analyze, we should recommend we analyze the high quality base calling.
Yeah.
Sí,
It's called accurate.
Super accurate. There's three levels. There's fast, there's high accuracy, and there's super accuracy.
creo que la van a enviar.
Did the copy say that as well? Just you should.
Yeah. Yeah. You can maybe on Thursday, you can share results for the Thursday meeting.
Next Thursday?
Yeah.
Oh, I see.
Sí.
Yeah.